Okay.
Yeah.
How about now? Is it okay? The slide?
Okay. Yeah, it's good. Yes, good to go. Yeah. Okay.
Okay, thank you, Dr. Karthik, for the invitation to join this Pyro Asia Forum bi-monthly webinar. So my name is Siti Baidurah Yusoff from University of Science Malaysia. So basically, my presentation today is titled, I will address the topic of biodegradable plastic, titled Rapid and Direct Compositional Analysis of Polyhydroxyalkanoate in Whole Bacterial Cell by Pyrolysis Gas Chromatography. So basically, this is just a definition of the biodegradable polymer. So basically, we can define this biodegradable polymer as a polymeric material that can be degraded or broken down by the microbes such as bacteria, fungus, enzyme, and other organisms. So basically, after degradation, it will produce a byproduct such as water and carbon dioxide. So this degradation process can occur in aerobic, meaning with the presence of oxygen, and without oxygen, anaerobic. So the steps of biodegradation of biopolymer, we can divide into three basic steps. Involve degradation of the outer layer of any kind of polymeric material in the presence of extracellular enzyme. So this enzyme will digest the outer layer of the plastic bottle or thin film in the shape of film samples to produce the corresponding oligomer. So this corresponding oligomer will then later will be absorbed into the microbes and again will be metabolized by the intracellular enzyme and produce water and carbon dioxide. So basically this is the general concept or theory of the biodegradation mechanism of polymers. So the polymers we can classify into three basic origin based on what kind of production process. So basically origin number one we can classify produced by bacteria. So in this group such as polymers such as polyhydroxybutyrate (PHB), polyhydroxybutyrate co-hydroxyvalerate (PHB co-HV), then gellan gum, then cordyceps. Another group is biodegradable polymer produced by natural products and their derivatives. So this is including amylopectin and chitin, and also cellulose acetate (CA). The third group we can categorize this biodegradable polymer produced by chemical synthesis such as polyglycolic acid (PGA), polylactic acid (PLA), and polycaprolactone (PCl), polybutylene succinate (PBS), and its copolymer polybutylene succinate co-butylene adipate (PBS) co-BA. So in this session, I will use this polymer of PBS co-BA, and another one is PHB co-HV, for example, applied in the analysis using reactive pyrolysis gas chromatography. So using this plastic sample, I will try to explain in a simple way how we analyze the degradation product. So basically, this is the schematic diagram of reactive pyrolysis gas chromatography. So inside this pyrolyzer, we have this how to say pyrolysis heater in this small inside this pyrolyzer. So at this part, the pyrolysis occur. The temperature we can set around 300 to 500 degrees Celsius. So in the beginning sample preparation, we take this small platinum cup, we put the polymer or any kind of sample of interest. So in this case, I will be using polymer and addition of tetramethyl ammonium hydroxide. In this case, we use 2 microliter. This is the optimized condition. The amount is 2 microliter. We put it inside this small platinum cup together with the polymer sample. So looking at this section, the pyrolyzer in detail, we put the small platinum cup on top of this section, initial mounting. So this is at this stage, it is in the room temperature. So when we insert, we press this button so that it will drop into this pyrolysis section. Okay, so once it is heated and it will transfer into the column of gas chromatography, then separation will take place and it will detect by this flame ionization detector FID. Okay, so basically this is the schematic diagram what happened during the sample insertion at this pyrolyzer. So the first sample I want to share is using this biodegradable polymer P3HP CoHV. So basically this polymer is accumulated inside the bacteria cells. We produce through fermentation of this kind of bacteria, Cupreavides nicata, C nicata. So we prepare this production of biopolymer. Then we subject this sample for the detection, direct detection using PYGC without any sample extraction or any kind of pre-treatment. So basically the experiment part, I will go in detail just for this slide in detail about the experiment part. What we do is the first stage of fermentation, we do something like how to say, bacteria proliferation. We increase the number of bacteria in this first step. Then using shake flask after overnight culture, we centrifuge, then we discard the supernatant. We get the cells. Then on the second step of fermentation, at this stage, we want to increase the production of the biopolymer inside the cells. We modify the media so that it contains limiting nitrogen source. Then in this condition, the bacteria will accumulate the polymer. We centrifuge again to get only the cells, bacteria colony. Then we wash with acetone 1 ml. Then wash again to get this dried powder samples. At this stage, these dried powder samples for the analytical part, we subject to two separate analysis method. One is reactive pyrolysis-GC. So this advantage is we can use within analysis within only 20 minutes. In contrast to conventional method, whereby we need to do sample extraction, it will take around 1 day for this analytical part using conventional methods. So I will explain in detail the conventional methods. What are the steps we do is from the obtained right bacterial cells, we do this depolymerization so that we can get monomer unit of HB and HV. So this one, we do transmethylation 100 degrees Celsius and 3.5 hours, then let it cool. We do the continuing pre-treatment of solvent extraction, whereby we add 1 ml of distilled water and 1 ml of hexane, vortex, and we leave it to get this two separated phases. The organic phase is further subjected for GC analysis. So this is the conventional method. Overall, it will take around one day approximately for the completion of the sample pretreatment. So in contrast, if we are using the reactive pyrolysis GC, it will take around less than 20 minutes without any sample extraction. So this step can be excluded if we use the reactive pyrolysis gas chromatography methods. Okay, so the results, this is the chemical structure of HB and HV. So it is in a polymeric form in a very long chain. connecting co-monomer of HB and HB. So using this formula calculation, we are able to calculate how many percent of HB monomer unit and how many percent of HV monomer unit inside the accumulated polymeric biodegradable polymer inside the bacteria. So here I'm sharing this two pyrograms. The top is the sample, standard sample, we call it, from industrial available sample, PHP equal HB. And the bottom one, the B, pyrogram B, is the one that we get from our analysis, reactive pyrolysis-GC, direct analysis from the cell culture without any kind of sample pretreatment. So we did not do any kind of pretreatment. We directly use for the analytical analysis using PYGC. So we can see here the the metric component derived peaks. So this one is derived from the cell outer layer of the bacteria. 1, 2, and 3, 4. This metric component derived peaks. So the rest, the 9 peaks, is derived from the monomer unit of HB and HV. We have successfully analyzed this biodegradable polymer, and we can calculate how many percent of HB unit and how many percent of HB unit inside the bacteria cells. So this one is basically in detail. If we have some time, I will cover this part later on. So I will skip this slide. Okay. So in comparison with the two conventional methods and the reactive PYGC methods, we obtain the data is almost the same. So we can see that this reactive PYGC, the HB monomer, is almost the same with the conventional. So this data we get RSD below than 5%. So this indicate the this indicate the RSD less than 5% indicate it is very high accurate or efficiency to use this kind of analytical methods. In contrast to conventional methods, because these conventional methods we need to do something like sample extraction and depolymerization. So by using reactive PYGC, we can directly analyze the bacteria cells without any kind of sample pretreatment. Okay, so this is another second topic. In different analysis, different experiment, what we do is we obtain this polybutylene succinate and butylene adipate plastic with the diameter of 30 millimeter, approximately 50 milligrams. So this circular plastic shape, we do this kind of soil burial biodegradation test. So we put this plastic inside the soil, inside the plastic container we cover, then we control the percentage of the humidity of the experiment, this setup. Then with the elapse of biodegradation time, in this experiment we do 4 weeks or 28 days of biodegradation. So in the first week, after the elapse of 1 week, we extract one plastic. Then with the elapse of the second week, we extract another one plastic. So following the third week and the final week is on the fourth week, 28 days, we collect the samples after each weeks of biodegradation. So what we observe is that this plastic is half portion of it is degraded and forming holes at the center and at the edge of this plastic. So at this local point of this plastic, we take the samples and we subject this by a degraded plastic film into the reactive pyrolysis gas chromatography analysis. So what we obtain is that this is the pyrogram. The top pyrogram is the one that we use without any reaction of tetramethyl ammonium hydroxide. It is quite difficult to analyze because it has many peaks derived on top of the pyrogram. But with the addition of tetramethyl ammonium hydroxide (TMAH) at the temperature of 350 degrees Celsius in the second pyrogram, we can clearly see these four main peaks. So the peak SD or peak AD is directly derived from this polymer chain, butylene succinate indicated by this red color, derived from this succinate SD. Then AD is derived from this adipic acid dimethyl. So by this copolymer composition analysis using reactive pyrolysis-GC, we can successfully indicate or monitor how many percent of monomer unit degraded during that period of soil burial degradation time. Okay, so basically this table is the summarization of the degradation time in days. So this is the 7 days, first week, second week, third week, and fourth week. So with the increase of degradation time, we observe the decrease in butylene adipate unit. So here the recovery percentage also decrease with the elapse of degradation time. So by using from this thin film, degraded thin film, using 20 micrograms of samples, we can analyze directly at the pinpoint, directly at the degraded section. So in contrast, this analysis is not able to be conducted using FTIR or any other conventional methods. So by using this reactive PYGC, we are able to analyze at the local pinpoint at which part of the film is degraded. We can analyze the copolymer composition directly. So this is one of the advantage for us to analyze using reactive PYGC. This is almost the end of my presentation. We received this pyrolyzer from Mr. Chu Watanabe. I believe now his son, Mr. Ichi Watanabe, is also actively handling the company Frontier in Japan. So this instrument is now in contract 2 years in loan to our University Science Malaysia. So this is Mr. Chu installing the pyrolyzer in our university in previous year, 18th of July. So with me, we are installing this pyrolyzer in this picture. Yeah, we really thank for the Frontier Lab for this loan of the pyrolyzer for 2 years to our university, USM. Yeah, I think that's all for my presentation, Dr. Karthik, for now.
Okay, thank you, Dr. Siti. It was a very wonderful sharing on this reactive pyrolysis application. Yeah, so there are some questions, so which we will discuss at the end. Now I invite Dr. Jain to quickly share about the upcoming Pyro Asia 2025 conference. So I request all participants to stay for another 5-10 minutes so that you can learn about this conference and also be part of the Q&A session. Okay, so let me invite. Yes.
So this is
Yes, it's good to go. Yeah.
okay. Good afternoon. I'm Jentila Latiwan from Thammasat University. First of all, thank you, Dr. Karthik, for having me here to share and introduce the Pyro Asia 2025 symposium that we're gonna have in Thailand. So I am the organizing committee of the Pyro Asia 2025 under the Professor Shanathip Samat as the chairperson of the conference. So the conference will be during 10 to 12 December in the end of this year. And we're gonna organize at Thammasat University Pattaya Center in Pattaya City, Thailand. So in the conference, this time we will have panelist speakers, the Professor Yoshioka from Toku University and Professor Trakung Witisat from Chulalongkorn University. Moreover, we have keynote speaker, Professor Pu and Professor Kumagai from Japan, also Professor Winmu and Professor Mohanty from India. In addition, we also have invited speaker from Malaysia, Indonesia, Canada as well. In this conference, we will share, talk about the experiment research on the topic of pyrolysis in both of analytical and applied pyrolysis. So, based on following topic or the abstract following this topic, I will come to be submitted. So as I mentioned that the conference will be at the Pattaya Thammasat University. So it is very close to the Pattaya city. If you travel from like outside of Thailand, so if you are able to choose the airport, so I recommend you to fly to Suvarnabhumi International Airport, as it is more convenient compared to Dongmung International Airport. So from the Suvarnabhumi International Airport, you can travel to Pattaya approximately 120 kilometers, which is less than 2 hours, and you can take a bus or taxi. And from now to 31 August, we are open for the abstract submission, and we send you the notification of the acceptance during around like September, and mid of October it will be like registration period, and the conference day, important day, will be like 10 to 12 December. In this conference, we also like giving the student for the best thesis award, and for the young researcher and also distinguished researcher. So this is the Pattaya city. There's a lot of tourist spot to join. So we are hoping to see you in the Thammasat University Pattaya campus for the Pyro Asia 2025. If you have further question, you can contact us through the email, or if you want to look up for more information, you can scan this QR code or visit our website webthammasat.com/pyroasia 2025. So thank you, and hope to see you.
Thank you, Dr. Jain. Yeah, so participants again, yeah, please consider joining Pyro Asia 2025 and